Okay. क्या नाम है? Prince Nagpal. Can you hear me? Mansi, can you hear me? Yeah, let me know, please. Okay, thank you. So today I'm going to use this platform, and I hope this is convenient to all of you. I will be monitoring how many of you are really joined in my YouTube account, and that will be the way I will be taking your presentee. Okay, I'm seeing all the names on my screen, and that way I will be able to manage. Okay. Okay. So, see, there was some issue with my Google account, and I am unable to stream my video. That was the reason why I have come through this particular platform. This is same as Zoom, and whatever you are posting in the chat box, I am able to see that. Okay, and I can see the names of each one of you. So after some time, I'll ask you to just give your presentee, and you will have to say yes, sir. That will be marking your presentee. That's all. Okay, that's the way we can go ahead. This is our second practical. In the first practical, we had talked something about microscopy. Those details we are going to finalize today. Then I'll ask you to go through the contents independently using your own mobile phones or laptops. After that, I'll be coming back again after 10-15 minutes, and then we'll be seeing what is your feedback, and we'll go ahead in that way. Okay. So friends, last time I had shown you, or I had discussed with you, some details about microscope, but we had not seen any videos or images or what are real parts of it. Okay, so that's what I'm going to do today. In the beginning, I'm going to show you a short video, small video on my screen that will be taking you through the parts of this particular microscope, which we have discussed last time, and then we'll be talking about the theory related to that microscope. Okay, I'm sharing my screen. So I hope you all can see my screen now. In this, I'll ask you again. Okay, is this video visible to you? Just someone answer whether this video is visible to you or not. Just a minute, I'm checking it. I'm running this video for a minute. Okay. I know you all are able to see this particular video. In this, initially, some information about microscope has been shown. Okay. So this is what we call as or compound microscope. There are various parts of it. See, this is how we have to put on the microscope and adjust the light intensity. Okay. Then this is the microscope stage where we are putting microscope slide. Then this is called as eyepiece nose or nose eyepiece. We have to just rotate this nose eyepiece and focus the lens on the specimen that is present on slide. Okay. Just a minute. We are going to see what is there in that. The video is little slow, but it will operate. No problem. Okay, just a minute. Just 1 minute. I'm stopping this video and I'll be sharing it once again so that it runs fast. Okay, just wait for a while. Okay, so we are doing it from here. The video is taking a little longer to upload, but I hope it will work properly. Just cooperate with me for a while. You'll be able to see it. 1 minute. Just 1 minute. ओके, समवन हैज सजेस्टेड कि आप लोग वीडियो यूट्यूब में देखेंगे. ठीक है. ओके, लेट्स ट्राई दैट ऑल्सो एंड आई विल बी साइमल्टेनियसली स्क्रीनिंग इट. दोनों करेंगे हम लोग. लास्ट टाइम आई हैड आस्क्ड यू टू विजिट अ वेबसाइट and that website was vlab.co.in. Okay, you have to log into vlab.co.in. You start doing this on your mobile phones. In that, yeah, I know the video is blurred because I think there is okay, let's not talk about issues anymore. You just go to this particular website, vlab.co.in. I'm showing you that website on the screen now. See, this is the website about which I am talking at present. I'm talking about vlab.co.in. Okay. If you go to this particular website, this will be the page that you will see. Okay. Hope you all are there on this page now. This is the front page. When you type vlab.co.in, this is the page which you will see. On this page, you have to go down and at this position on biotechnology and medical biology, medical engineering, you have to click. When you click here, you will be directed to another page, which is this page now. Okay. On this page, you have to go to bottom to Cell Biology Virtual Lab 1. And then on this Cell Biology Virtual Lab 1, you get a link to this microscopy, light microscope. Right? Here you have to click on simulator. You will be taken to that video now. Okay, that particular link will take you to the video related to microscope. Okay, someone is saying that YouTube is consuming too much data. I'll have to see if YouTube is consuming too much data, then you have to reduce the resolution of your screen. You make it minimum resolution. Okay. And this is where you will be getting the video related to microscope. Okay. Reduce the resolution of your YouTube channel and then see this particular video. This is what you have to do now. Okay. It is a 2.5-minute video. You see that and we will be coming back to the discussion again. Okay. In biomedical engineering, you have to go to Cell Biology Virtual Lab 1. Cell Biology Virtual Lab 1. In that Cell Biology Virtual Lab 1, there is second experiment of light microscopy. Shaloni, I accept that this is inconvenient, but at present we have to deal with the content like this only. I'm trying at my best to streamline this particular process. It will be done today. Okay, let's just see this one part first. You have to just observe the video of microscope here, nothing else. Okay, I accept it is first practical. It is going to be little inconvenient to you. Accept it, but be there with me for a while. let's try to sort out this particular problem. Okay. So in this particular video, you will be able to see what all are the parts of a microscope. Okay. I hope by now you have solved the problem of data consumption. You have reduced the size of your or resolution of your video. Then I had asked you to go to vlab.co.in. In that vlab.co.in, you have to go to biotechnology, biomolecular engineering section. There you have to go for cell biology lab. Yes, and then you have to click on animation. Click here for animation is link there. On that link, you have to click and you have to watch that video where you will be getting a virtual presentation of what are contents of or what are parts of a microscope. Simple. Okay. This is just 2.5 minute video. You observe that video and come back to me. I am giving you 5 minutes gap here. Okay. Okay. There is some concern about ये practical file में लिखना है क्या? Okay. लिखने से अच्छा है आप लोग print निकाल सकते हो. You can take a printout of this and you can put it in the file. That will be fine. Okay. That resource I am going to show you immediately after this video. Once you see this video, we'll be coming back to another page of this particular website where I will be telling you theoretical aspects of this, and then we can come back to how to write this practical. There is a content, ready-made content. You have to take printout of it and place it there. Okay. Just I'm waiting for a couple more minutes. By that time, your video will be over, and then we'll go for further details now. Okay, majority of you have finished seeing that video. Now you can pay attention to me. Okay. If all of you have seen the parts of that video, parts of microscope, then you can pay attention to me. Say I'm starting with the further discussion now. Okay. Okay. Regarding this written experiment, this is another website to which you have to register. Okay, that exercise I am going to ask you to do after this particular demonstration. Okay. Who has asked that question? Ritika Gupta, you asked me how I have come to this particular website, right? I'm going to tell you after the finishing of this particular explanation. I will tell you how to reach this particular website, okay? And that you can try to access and get the details from that. But that we will do after finishing this particular explanation. Is that okay? All right. So now pay attention to me here. This is another website for virtual practicals, and few practicals of your syllabus are covered in this particular website. Okay, don't try to visit this website at present, just pay attention to me. We are talking about the first experiment, that is light microscope. The purpose of this particular experiment in your syllabus is that you should be able to describe various parts of the microscope, and then you should be able to independently use a microscope for performing your routine experiments like blood cell count. Okay. In this semester, you will be learning regarding WBC count, RBC count, and differential count. These three practicals we are going to perform when you get a chance to come to lab physically. Before that, we have to understand some details like this, and these are static details. Your handling of instrument is not expected at present, so we are doing this particular thing in the present mode. Okay. Now, once we have understood that the purpose of this practical is to find out or know what are various parts of microscope, we have to see about some history of these microscopes. Okay. You must be knowing regarding a name of Antony van Leeuwenhoek. Okay. Antony van Leeuwenhoek was a grocery shop owner of 16th century. Okay. And just out of interest, he had devised a simple microscope in which he had used lenses, couple of lenses he had used, and using those lenses he had prepared a very simple microscope through which he could see the magnified images of some biology samples. So this happened around 400 years ago. Okay. By using lenses, the shopkeeper had prepared a simple microscope in which only one or two lenses were used, and he could magnify the samples by few magnifications. By few times he could magnify the objects. That was the history about him. After that came the concept of compound microscope. Now the difference between simple microscope and compound microscope is that in simple microscope you are seeing actual image, means you are focusing that particular simple microscope on an object, and you will be able to see an enlarged image of it, and that is actual image, that is live image. Okay. But then there is another kind of microscope which is called as compound microscope. In this compound microscope, the object is magnified, but the image that you are seeing is not the exact image or live image as you see in simple microscope. What you see in compound microscope is a virtual image. Means once that image is magnified slightly, again that magnified image is magnified once again by another lens. That is the arrangement in compound microscope. That is the reason why we call it compound. In simple microscope, live image you are seeing. In compound microscope, you are seeing a virtual image because it is magnified twice. That is the reason why we call it as compound microscope. Okay. That is what is explained in this particular first paragraph, that the first magnified image obtained with the objective lens is again magnified by the eyepiece to give a virtual inverted image of the object or of the sample kept below the lens. Because of this particular aspect, we are calling the microscope as a compound microscope. So this is the difference between simple microscope and compound microscopes. In simple, you are seeing the image directly. You are seeing the object directly through one lens or two lenses. But in compound microscope, one image is created and that image is again magnified. So that virtual image you are seeing in a magnified manner in compound microscope. This is the major difference between these two. Okay. Now again we are seeing the same thing, that there are various parts of microscope. Just now you have seen the video. I hope this is not irritating you now. Don't get irritated. This is the first and simplest practical. Last time I could not stream all these things on the screen. That is the reason why we are repeating it. Otherwise these are simple things, I know. But today we are going to see a couple more added details about microscope here. So friends, we have seen what is base of a microscope, what is C-shaped arm, and then there is mechanical stage which we can either heighten or lower, raise or lower, or there are microscopes in which the lenses will be lowered or raised. Okay. Both kind of arrangements are there. Now, apart from these mechanical parts, there is more important part, that is magnification related part, magnifying parts. There are two magnifying parts in compound microscope. One is eyepiece, another is objective. Okay, so last time we have seen regarding this, that objective is having some power and lens is having some power. So when you want to calculate the magnification, you will have to multiply the power of objective with the power of lens. That is that simple. Okay. So this is about the magnifying part of microscope. Then there is something called as illuminating part. Okay. About this we had cursorily discussed last time. Just I had mentioned that there is a part called as condenser, there is part called as diaphragm or iris diaphragm. These two parts are considered as illuminating parts for the specimen. Why we are calling them as illuminating part? It is because this condenser is present just above the light source. Okay. Condenser is present just above the light source, and the function of this condenser is to collect the light and focus it on the specimen. Now where the specimen is kept? Specimen is kept on the stage below lens. Okay. And this particular condenser, it will collect the light, it will diffract in a particular angle, and it will be focusing that light on the specimen. That is the work of condenser. With condenser, you are illuminating the sample kept on the stage. Illuminating means you are making it brighter. Is that okay? How you are making it brighter? By collecting the light and focusing the light on it. This condenser actually contains a convex lens. Okay. This condenser contains a convex lens. And because of this convex lens, the light which is coming from light source, it will be focused on the sample which is present just below the lens. Apart from this, to control the light, there is another part called as iris diaphragm. Now, just before some time, in the video you have seen that there is something called as iris diaphragm, and you have to adjust the intensity of light by moving the knob of this diaphragm. You might remember that in that video it was mentioned that when you come for high power lens, just open the iris by 2/3. This was the description which was given in that video, right? Means this iris is helping you to adjust the light intensity, whereas diaphragm is helping you to illuminate and focus the light on the sample. Okay, these two are helping as illuminating sources, and you have to keep on adjusting the intensity of light and level of condenser when you are using different power lenses. If suppose you are using 4x lens, then generally you have to keep light at minimum, illumination at minimum. Means condenser at the lowest possible level. When you keep condenser at the lowest possible level, light will not be focused on the sample. Okay. When you go for 10x or 40x, it is necessary that to visualize that particular sample properly, you need to illuminate that particular objective or particular sample. To illuminate that, then you will be raising the level of condenser so that light will get diffracted and it will be pointed towards the specimen, towards the sample. And then you will have to open the iris diaphragm so that intensity of light also is more. So by using these two things, you will be adjusting the illumination, brightness and clarity of the sample specimen that you want to observe. Then there is something called as light source. Now there are various types of light sources. Monochromatic light sources are there, or simple tungsten halogen lamps are there. These various kinds of light sources we can use dependent upon our need. Now, why I'm saying our need? Because sometimes you may need monochromatic light, that too in a particular angle. Why? If suppose you are interested in seeing crystals, shining crystals which are present in your sample, then it is necessary that you have to polarize the light, is make the light to pass in a specific angle, specific plane, and then that light should be projected on the sample. That kind of microscopy or that kind of light source we call as polarized light source. Okay. What is significance of polarized light source? It polarizes the light coming out of light source and focuses the polarized light in a particular angle on that specimen, so that if there are any crystals in the specimen, those crystals will shine and you will be able to observe those shining crystals. Okay. Sometimes this polarized microscopy is necessary. That is where you will have to have a proper filter or a proper light source for that purpose. Otherwise, general LED lights also we can use, right? Then by now you have seen coarse adjustment, fine adjustment, those things are there. You can check them. You have just seen them, how to adjust the specimen or focusing by using those fine and coarse adjustments. Last time I had also told that in further advanced microscopes, these fine and coarse adjustments are having markings on them. Okay. I'll try to see whether we can get such an image where you can see scale on the fine and coarse adjustment of microscope. going for images. I'll check whether I can get any image where scales have been shown on this particular parts. Okay, here we get it. Just a minute. One minute, that image is coming up now. Just wait for a couple of seconds. The image will appear. So what we are talking about here, it is regarding the scale which is given on fine and coarse adjustments. Here this site is taking a little longer. Can you see it on this particular part of your screen? See here on this fine adjustment knob, you can see some scales are there. Okay. I had asked you why this scaling will be necessary, right? And then I had mentioned last time that this scaling is similar to screw gauge or vernier caliper related scaling, using which you will be able to see through the depth of the tissue, or you will be able to define how deep you are looking into the tissue. That is the purpose of this scaling which is given on fine and coarse adjustment. Okay, let's go back to our slide, same slide again. Now this particular diagram shows us how actually an image is created in a microscope. Okay. So we said that there is something like light source, that will, then there is diaphragm, then there is luminous field, means condenser. Through all these setups, the light will go and it will fall on the specimen. Then a immediate image plane is there, where a virtual or where a copy of image of this particular specimen is created, and then that virtual image is magnified and we can see that image here. This is how the entire process of image generation occurs. Okay. So just now you have seen the details of this particular text, where you have seen how to adjust the video and how to see the image. Then in last practical we had also discussed about how to, or what is the amount of magnification that we get by using different permutations and combinations of lens and objective lens, or eyepiece and lens. Okay. If your eyepiece is of 10x power, lens is of 4x power, you are just getting 40x magnifications, 40 times magnified image. Okay. Then this is the new concept which we have not discussed in last practical, and the concept is of resolution of image by using microscope. Okay. To calculate that resolution, now how we can define resolution? Resolution is ability of the microscope to differentiate between closely lying two objects. For example, if suppose you want to differentiate the gap between nucleus and cell membrane, whether your microscope is able to do that or not, or how much minimum distance your microscope can differentiate. That's what you have to understand in the terms of resolution. Understand resolution, you should also know what is meant by numerical aperture. And numerical aperture is something, this is numerical aperture, that is dependent upon the angle with which the light is diffracted, the light is scattered. Okay. And then by using this particular formula of 0.5 lambda divided by n sine theta, you can get resolution of your microscope. Okay? And in calculation of this resolution, you are actually using this parameter, numerical aperture of objective, numerical aperture of lens. Okay? And this is how you calculate the power of your microscope to resolute two atoms or two closely lined substances from each other. Okay? So these are the details about compound microscope. Now within couple of minutes, I'm going to tell you something about how to get registered to this website. I'm telling you how to go to this particular website. For that, see, we have to first type here VLab amruta.edu. Hope you all can see this. What I have typed here is vlab.amrita.edu. You click it, you will be taken to this particular web page. After going here, you have to log in. And for this logging in, you will have to, when you're first logging in, you will have to fill up your details. Okay. For logging in, you'll have to fill up your details. You put your name, your email ID, your college name, whether you are a student or not. All these details you have to fill up, and then go for registration. After registration, you will be getting an authentication mail on your email ID. So I hope by now you all are having your email IDs. So that email ID, you will be getting an authentication code. Click on that code. You will be again coming to this particular website. Here you can get the contents of microscope and your first experiment of cell counting. Okay. Again, you need not to worry too much if you are unable to go up to here, or if you are unable to register to this website. No issue at all. Okay, I'm going to share all these details with you in your WhatsApp groups. There is no problem at all. Don't worry about anything. Don't get panic. These things you are going to do very easily within a couple of days, within a couple of hours, and within few minutes also you will be able to do that. So you have to try this now. I am taking break for next 15 minutes, within which you can try all these things once, and I will come back to you with further details now. I will join back at 4:10. 4 hours 10 minutes I'll join back. Still then you go through all these details. We will meet after 15 minutes once again. All right. You have to again come here on the same video link. I will be joining after some time. Okay. Be there up at 4:10 to 4:15. At 4:15 I'll be there. Okay. All right. So try this. We'll see how far we can go. Okay, I'm waiting for others to join. Let few others join that so that we can start discussing again. Okay, there are around I think 39 students present here now. Okay, I'm going to share my screen once again now. And we're trying to have look at further details of it. Okay. I hope you can see the screen. On the screen, I have opened the same website again. And in this website, we're going to see a small assignment here or self-evaluation here. Okay. Now I want you all to interact with me through this YouTube chat, and let's try to see whether we can solve some simple questions related to microscope or not. Okay, I'm going to self-evaluation button of this. I hope you all can see what is visible on the screen at present. And I want you to read the first question. Okay. Just now, whatever discussion we have done, dependent upon that, there is a question on this microscopy. This is related to simple and compound microscope, where the first question is the image produced by compound microscope is. There is option A, real magnified image, B, virtual image, C, real diminished image, and fourth, D, virtual diminished image. Just you have to tell me whether the answer is A, B, C, or D. As many as you can understand this question, you just go on clicking whether it is A, B, C, or D? Just to tell A, B, C, D. That's all. A, B, C, D. Right, majority of you got it correctly. It is virtual image. Okay. Few of you initially answered it as D, but the correct answer is virtual image, because this is the basis on which the microscope has been named as compound microscope. If it had been a simple microscope, the image could have been real magnified image. Real magnified image comes in simple microscope, not in compound microscope. This is virtual image. It is not the diminished image. The image is there. The image is being more clearly shown. Okay. Okay, someone is saying that I am not visible or audible. Is that the problem? I think everyone is able to see and hear me, right? See, you will have to just check your own audio. Who has said that? I think someone has said that I am not audible, visible. Okay, Saloni Sharma, you have to just check whether you have put your phone on mute mode. Okay, others are able to listen to me. So this was the first question that we have taken up. And we have seen the difference between simple microscope and a compound microscope. I am going to next question now. Hope this next question is visible to everyone. The next question is 4x objective lens is also called as, it is called as low power, high dry power, oil immersion, or scanning. Again, you have to answer in terms of ABCD. 4x lens, what it is called as? Low power, high power, oil immersion, or scanning. What is the answer of this second question? Can anyone answer this? Okay, exactly. There are two answers I am seeing on screen. That is either it is A or D. Okay. See, actually the correct answer is A. We call it as a low power lens. Okay. We call it as a low power objective lens. Right. It is not scanning lens. It is objective lens. All right. We have finalized second question. Let's go to third question now. Third question is, when do we use only fine adjustment knob to focus? This is the question related to that video which you must have seen just in the beginning of this particular practical. Here the question which is asked is about operation of that microscope, and it is asked when actually we should use only fine adjustment and not coarse adjustment. Is it for oil immersion objective or for low power objective, or there is no such rule, or when you are using scanning objective, in which case you have to use it? Yes, for this majority of you are getting it correct, that you have to use only fine adjustment when you are using oil immersion objective, because what happens is while using oil immersion objective, always there is problem that the lens will get damaged if you use the coarse adjustment. Okay. So the answer is A. In case of oil immersion, we have to use fine adjustment, Because see what practically we are doing is, while setting this oil immersion, what we do is we put a oil drop on the specimen, dried specimen, and then we turn the lenses, or what we call it as nose lens. We bring the 100x and we touch that oil drop with 100x lens. Here we are touching the lens to the sample specimen which is in oil, so that oil is actually touching the lens. Means there is very small distance between the sample and the lens. If suppose forcibly you at once move the course adjustment, then that movement may damage the sample as well as it may damage the lens also. Right? For that reason, if you are using oil immersion microscope or oil immersion lens, oil immersion objective, you have to very carefully use only fine adjustment. Okay. Even in practicals, we tell this as a rule for high power lens also. Okay. For high power lens also, the distance between sample and objective is very small. For that reason, to avoid damage or avoid the sticking of that specimen to the objective lens, we have to use only fine adjustment. Then there is a tricky question. Very rapidly we had gone through these details of resolution of microscope, limit of resolution. So let me tell you, in case of our compound microscope, routine laboratory compound microscope, the resolution power is 2 micrometers. What is resolution power? It is 2 micrometers, 0.2 micrometers. It is 0.2 micrometers. Now, when you convert this 0.2 micrometer to nanometer, it comes to 206 nanometers. Okay. So, for compound microscope, approximately the resolution power, limit of resolution, is 0.2 micrometers or 206 nanometers. That is the limit. Right, the answer for this is B. Right. And then there is one more question. To visualize that, I will have to reduce the size of screen. Okay. Just a minute. I'm refreshing the screen again. All right. So first question we had said it is virtual image. Second we had said it is low power image. Third is while using oil immersion. Fourth is 206 nanometers. And then there is fifth question on the screen. That is parfocal refers to microscope with multiple objectives. If suppose one microscope is having multiple objectives, it is called as parfocal only when just a minute, it is getting disturbed. A microscope is called as parfocal, or observation is called as parfocal when there are multiple objectives, but each objective is positioned at the same stage height as that of others. Okay, if that kind of arrangement is there in a microscope, we call it as parfocal microscopy. Okay, let us check whether these answers are correct or not. Okay. What is for low power? Okay, now we have seen some details regarding this. Let me know whether you were able to log into this slide or this particular site or not. Okay, yes, Forex is called as scanning, that is right. Forex is called as scanning. All right, see, let me know now whether you were able to get into this particular website or not. How many of you could register for this website? My question is, how many of you could register to vlab.amrita.edu? I want to check how many of you could do that. Okay, are you getting access to this? There are so many who have done that. Okay, that's great. That's great. So all of you are having access to this particular website now, right? Others who have not done, you go and do that. Solve this particular self-evaluation question. You can see the animation here. You can see simulator. You can see some further references also. Okay. There are other details. There are book names. There are websites from which you can get further details regarding this particular website. Okay. So all these details you can visit and read today. There are important links to history of microscopes. Okay. Who discovered it and all other details are there. Just now we have talked about Anthony van Leeuwenhoek, Robert Hooke. These are the scientists who did this. Now these extra readings are there just for knowledge purpose. If you can remember it, it's okay. If you cannot, even that is okay. No one is going to ask you questions on this. Okay, just for sake of knowledge, just for understanding purpose, we want all these details. Otherwise there is no problem. Okay, all right. Now let's come to the first question again, whether to write these details in the journal or not. Okay, for that the answer is you can just copy all these things, paste it in a Word file, set the pages properly, take it print out and add to your journal. That's enough. Like this, you have to select it and then copy it, paste it in Word file, and take a printout of it, put it in your journal. That is for first practical. Okay, you need not to write all these things again. It is not necessary. Okay, whatever contents are there, you can just bring them to Word file, paste them here, and finish your practical. Right? There is possibility that you will be asked to draw a diagram of microscope in the exam. So on a page, you can draw an image of microscope. That's all. See, you need not to write anything. Whole content is easily copied. You can paste it into Word file and you can save it as your first practical. Is that okay? That is how you have to finish your first practical. There is no need of writing these contents again. You just keep this thing saved as practical number 1 with you in your mobile or in your pen drive. Whenever you will be coming to laboratories physically At that time, we will be asking you to prepare a file of these documents. You can paste these documents into that file. That will be your experiment number 1. Okay. Journal, that's I'm telling you. Journal means at present you prepare this Word file, label it as experiment number 1 APHE, save it with you in a pen drive on today's date. Okay, today is 16/12. So on this date, for the practical, you have to keep this as the content. You remember this website, go through the details once again. There are details like how to operate this particular microscope, what care we have to take, how to maintain the microscope. Okay, there are some assignments associated with this. You just go through this, read this particular website. And here we finish our first practical. Okay. I know that because of initial jerks related to joining and initial problems related to streaming, there was some nuisance in the beginning. I promised you that I will try to solve these issues, but somehow I could not do that. I will have to buy a camera now, webcam I will have to buy, and then I will have to check whether it really works properly or not. So I will do that in coming week. I will have to buy a webcam. My webcam is not working. What is the problem? I do not understand. But then we'll be doing this by next week. Right? So is there any question about this particular detail now? If there is any question, you can post it in chat box. Otherwise, let's finish this practical and let's go to introduction of the next practical now. All right, this was very simple experiment. I can understand little bit, it must have been a kind of repetition to you all. The things were very simple, but mind well, if you do not attend to these details, sometimes it becomes difficult for you to use microscope at later stage. Okay, so it is important that even though the things are simple, you have to look at them little more carefully once at a time, and then it will be all right. You will be able to manage working on microscope once you get a practical hand on it. Okay, all right. Now the next practical that we are going to perform is of hemocytometer. I hope you all can see this on screen now, of your mobile screen now. The practical that we are going to do or understand next is hemocytometer. Okay. Now, this is something which we use to find out number of cells per ml of blood or number of cells per cubic mm of blood. Okay, or number of cells per 10 microliter of blood. That's what we are going to do in this next practical, that is a cell count. Saloni Sharma is saying end it for today. Okay, there are 10 more minutes which are required. Saloni, can I understand why you want to end it earlier? Just I want to know. Okay, someone is asking whether you have taken attendance or not. All right, from Saloni, I want to answer why you want to end it earlier. Okay. See, attendance is what I will be seeing all your comments. Just when I started now, I had asked everyone to say yes, sir, and majority of you have said that yes, sir here. So whoever is present, I will be just counting this yes sir as a today's presentee. Okay. So if you have not said yes sir in the entire session, then just there is a request that you have to say yes sir once again. Okay. Saloni, you please bear with this today's problems. See, we are trying to resolve these issues. Sometimes it becomes problematic for us also to try. We are trying at our best to serve you better. Okay. So let's see how better we can go now. All right. I'm going to just give you a brief introduction of this practical and then we'll finish it. Okay. I'm just showing you some image on the screen now. This particular image. I hope you all can see this image. What you can find on the left side here is a slide, which is also called as Neubohr's chamber. Okay. This slide is called as Neubohr's chamber or Neubohr's slide. Okay. On this particular slide, you can see there are particular type of grooves available here, and you can see a H letter like something is carved on this particular slide. Okay, I hope this is visible to this. Hemocytometer is the second experiment, right? We are going to talk about that hemocytometer only. So this is a glass slide which is very thick as compared to your routine glass slide, and on this glass slide there are grooves. A deep carving is there. This one line, the second line on the other side, then this is third line, this is fourth line. So these four grooves are there, and because of that these two raised parts have been created. Okay, and then at the center again there is one horizontal groove. Now because of this, what has happened is there are these two raised parts, this third raised part and fourth raised part. So these four distinct raised structures are created on this slide by creating these grooves. What is significance of this? You are actually going to place a cover slip. I hope this cover slip is visible to you. This cover slip you are going to lift and put on this particular area here. Okay. When we put this cover slip on this area, what happens is this central two raised portions which are there, these raised portions will still stay lower than the cover slip. There will be a gap between the cover slip and these two central areas. Okay. And it is going to have some significance in further calculation. So these grooved parts we are saying are lower than the cover slip surface, lower cover slip surface, and there is a small gap between cover slip and these two raised parts. Now, when we see this entire Neubohr chamber slide under microscope, we can see fine groovings on them. Fine, you can say very finely and neatly placed lines are there on this particular Neubohr chamber. You can see these lines on. Okay, someone is saying Saloni is not able to see anything. Okay, let it be, Saloni. You just keep on listening. There is no issue. Okay, no issue. You can yourself visit this website again and you can see all the details. Just we are going to get acquainted with what is Neubohr chamber, and we are going to finish this particular discussion here itself. Right. So there is grooves. I told you cover slip if you put, it will be having some distance between this raised part and the cover slip surface. On these raised parts here and here, very fine engraved lines are there. and these lines we are going to use for enumeration of cells. Okay, someone is saying that I will have to share it once again. ठीक है, ट्राई कर रहा हूँ, 1 मिनट रुकना आप लोग. Okay, let's hope that you are able to see this. Up till now, I have told only 2 or 3 details about this. On this Newberg chamber, there are raised areas here. 4 distinct raised areas are there. Out of them, these 2 continuous lines which are there parallel to each other, these are maximum raised surfaces. As compared to that, the central 2 surfaces which are again raised are slightly lower than the level of these 2. Because of this, when you put a cover slip on this particular raised part, a small gap will remain between the surface of this raised part and the surface of cover slip, lower surface of cover slip. Means between these 2, there will be a small gap. Okay? That's what I am trying to tell you. And then if you see this particular slide, this portion under microscope, I am telling you that it is having very fine linings engraved on the surface of this particular slide. Okay. So such slide is called as hemocytometer or Newberg's chamber. Okay, the name of this particular slide is Newberg's chamber. And we use this for enumeration of counting of WBCs, RBCs, or any other cell suspension. Okay. So with this brief introduction, let's stop here. By next time, I will try to get something that will be streaming the entire details with speed. Maybe there was some issue with today's college internet also. We will try to solve it. All of you get registered to this website of freelab.amruta.edu. All of you also visit freelab.co.in. These 2 things we have to do today. Okay. freelab.co.in is what you have to visit. And next is, it is not FLab, it is VLab. I don't know why it is coming. VLab.co.in. Okay. This is one website that you have to visit. Another is vlab.amrita.edu. You have to get registered to the second website. These 2 things you do today. And let me know about this. If there are any issues, you can send those issues to me on WhatsApp, my personal WhatsApp. Don't share it on college WhatsApp. You share it with me on personal WhatsApp. Whatever was your experience, whatever was your problem, complaint, you can send it to me on my personal WhatsApp. Okay. So next time we will meet again on Saturday. We are going to meet with one more topic. That will do. Okay. I'll share the timetable for that Saturday's lecture again. I'm ending this broadcast now. We'll meet again.